Okay, so what I will do today, and maybe before I share my screen, actually, I will go to the program page where you can kind of see that we structured this program around the Q&A sessions. The first part of the program is going to cover the introduction to the basic skills that you must have to work with genomic data. That will include a brief overview that we will do today of all the tools and different aspects that you can learn about. Then you can practice independently how to do these things on your own and make sure that you know how to do this independently. And then we will have a Q&A session on February 21st. And when we do that Q&A session, we are looking for your questions. What are some of the things that you didn't understand? What are some of the things that you want more clarity on? What are some of the additional topics that we didn't cover so that we can make sure that those are clearly outlined in this live Q&A session? And also, by the way, during today's session, feel free to post any of your questions or comments in the chat. That will give us a better understanding of what we should be covering and how to address the specific concerns that you might have. So what I will do today is I will give you a very quick introduction to genomic data processing and analysis. Then we will do some hands-on exercises so that you can get a sense of what are some of the different methods and tools that we will be using throughout this program. And then at the end, I will kind of go back to the schedule and some of the programmatic aspects of this training so that you are comfortable with the program outcomes, what is expected from you throughout this whole program. So the genomic data analysis program is designed to introduce you to the tools of analyzing DNA code sequences and making interpretations based on that analysis. And to begin speaking about this, let's briefly talk about next generation sequencing. So DNA was described, the structure of DNA in the 1950s, and then over the course of the next several decades, this rapid pace of technology development led to cheaper sequences, cheaper sequencing, and an abundance of data that could be analyzed that is used as a reference, that we know kind of what's happening in different diseases and in different populations, but also allows us to understand different specific project examples without even having to do sequencing ourselves. So we can actually take sequencing data from other labs, other research studies, and ask questions about DNA variation. But omics data doesn't just describe DNA. Omics data could refer to different types of omics. For example, phenomics, genomics, epigenomics, transcriptomics, even metagenomics, right? And so there are many different omics data types. And what we will do in this program is we will specifically look at genomic data in association with these other types of omics. For example, how does the genotype affect the phenotype? So how can we connect the morphology and the way that tissues and cells function with DNA sequences? Or transcriptomics, how are those defined by the specific genes that are expressed or the isoforms that are being expressed? So one of the things that we would like to understand is the relationship between structure, function, maybe evolution, right? How did we get here? And the way we can do this is by looking at known sequences. For example, genes that have been annotated, we can ask questions about what these genes do, or what kind of variation in the gene sequence doesn't let them do what they're designed to do. And then we'll talk about how the next generation sequencing data that describes the genomic variation can be analyzed to identify some of those changes, some of those variants. So essentially what we're looking for is, if we are thinking about the DNA code as a specific instruction about a protein or about a gene, we are looking for bugs in the code that change how that protein or that gene works. And a good example of that is described here with the red blood cell. You have the normal red blood cell, or you have the mutant, and that leads to disease. that is a result of a variant. So to understand where these variants are found and characterize them, we need to know a little bit about what types of variation we expect to find. In the DNA code, we have nucleotide sequences. You will often hear the terms base pairs or nucleotides, and those refer to individual nucleotides on one strand of the DNA, or two strands if it's a base pair. And essentially, those are organized into trinucleotide codons. Those codons code for amino acids, and amino acids are what the proteins are made of. So when we look at the DNA code, we're oftentimes thinking, how are these nucleotide sequences encoding for amino acid sequences, and how do the changes at the nucleotide level result in either a different type of an amino acid, or even a stop codon, right, that the whole sequence stops being transcribed at all. And so those are point mutations. Point mutations are going to take up the majority of our time. We're going to be looking at individual single variants that could either be categorized as substitutions, insertions, or deletions. So we can either expect one nucleotide to be substituted by another. We can envision we can encounter a nucleotide being inserted in the sequence or being deleted from that sequence. And based on where exactly in that codon sequence this change happened, we will expect very different types of outcomes. And those will be specific to different types of applications, because we will look at how germline mutations, somatic mutations, or even frequency of mutations in specific positions can have different effects across different conditions, diseases, and phenotypes. So the basic way to annotate and identify how this particular change can impact whatever happens at the phenotypic level or at the functional level, we can look at single nucleotide changes and describe them as those that occur in less or more than 1% of the population, and those that have more than 1% of the 1% of the population would be considered polymorphisms, or SNPs, single nucleotide polymorphisms. Then, if they are single nucleotide polymorphisms, we will consider whether they are in the coding or the non-coding region of a gene. And if they are in the coding region, we typically will ask the question, do they change the amino acid or do they not change the amino acid? And if they don't change, we will still think that maybe they could be associated with a phenotype, but most likely they don't change the structure of the protein. And even if they're in the non-coding region, we will also expect them to have some potential impact, especially if they're found in the promoter region or some splicing region of that gene. And then we will take a look at some examples that can help us understand how does this actually work in real life situations. So what types of mutations are there? Again, we have the DNA sequence, missense mutations, nonsense mutations, and frameshift mutations. And those are the basic types. So a missense mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene. Nonsense mutation is a change in one DNA base pair that prematurely signals the cell to stop building a protein. This type of mutation results in a shortened protein that may function improperly or not at all. Frameshift mutation is an addition or loss of DNA bases that changes a gene's reading frame, and the resulting protein is usually non-functional. So now that we understand this genomic variation, we can start thinking about how can we identify such individual variants, or what other types of variants can we expect to find. So if you imagine, and we'll talk about sequencing in just a few more minutes, if you imagine a sequence that comes out from a sequencer, typically we refer to that as a read. That read can contain a single position that has a change. If we align multiple reads to each other, many times we will find that one position has multiple changes. And so then we want to ask the question, are these changes consistent? What is the majority of the changes? What is the smaller proportion of the changes? And then we try to categorize them. And so that categorization can lead us to determine whether this is a single nucleotide variant, a single nucleotide polymorphism, or are we seeing other bigger rearrangements that we should be paying attention to? And those could include copy number variations. And ultimately, all of these could result in a change in a phenotype. So how does bioinformatics help us in this kind of genomic characterization of variants? Well, we will first need to understand how sequencing works to appreciate the need for computational analysis of genomic data, and then to actually practically see what does this data look like. So what I'll do in the next few slides is I'll talk about sequencing technologies and how they differ from each other, and briefly jump into an example of one of these technologies. And then we'll talk about several examples, and I'll actually use just one example today, and that will be an example from cancer. Next generation sequencing, as I already mentioned, has evolved dramatically over the past couple of decades. And originally, it started from a technique that was very expensive and labor intensive. And eventually now it is becoming more and more automated, as well as cheaper and more accessible. And so as a result, we actually have several different categories of sequencers. These are called short read sequencers or long read sequencers. Those are the dominant types of sequencing technologies today. And these are all next generation sequencing or the second generation sequencing technologies. The first generation sequencing technologies that were much more expensive are still used for a lot of validation. But primarily when we deal with this kind of data, we're going to be using these next generation sequencing sequencers. The machines are called sequencers. So here you can see Illumina machines. I think you have the Ion Torrent here, as well as Oxford Nanopore for long read sequencers, and PacBio or Pacific Biosciences. So the short sequencing technologies are the cheapest ones. The benefit of the short reads isn't that they're short, but that they can be very inexpensive, and they can be generated in a massively parallel manner, with billions being generated in a single run. So the cost is lower. And so when you need the cost to be lower and the count of reads or your ability to cover a certain region with a high number of reads is needed, short read technology makes the most sense. Long reads, on the other hand, can resolve regions of the genome that are inaccessible to short reads due to repeat sequences. And so if you have multiple sequences that are exactly the same, you're going to run into a challenge of mapping. And an alternative route is to generate longer reads. So the Pacific Biosciences technology can generate reads that are over 15,000 bases long. And some could be even over 100,000 long. At Oxford Nanopore Technologies, the platform, under the right conditions with high quality DNA, can produce reads of up to 1 million bases. So these are very, very long reads. And recently you might have heard that these technologies have been used to remap the human genome and identify a lot of the repeated sequences and actually determine what is in them. Now, these types of sequencers could also vary based on what is it that we are feeding into the machine. And that is what's called library preparation. So we can either sequence everything by just chopping up the DNA and putting it into the flow cell, or we could amplify specific regions and be selective about what are we actually trying to sequence. So based on this, when it comes to genomics, there are basically several approaches, and this is whole genome, whole exome, or targeted sequencing. When we talk about whole genome sequencing, we're typically expecting a fairly low coverage. The average is about 5 reads in a position, which means that we will, typically have, if we're again going back to those single nucleotide variants, right, we're going to be able to detect 5 reads that either all have a change in the sequence or only a proportion of them have that change in the sequence. So that doesn't have a lot of data to work with per each position. But the number of nucleotides is going to be huge. So if you think of the human genome, which is about 3.2 billion nucleotides, that is going to give you 3.2 billion positions with 5 reads that you can kind of measure what types of variants are you finding. The typical approach, though, is going to be whole exome sequencing. So whole exome sequencing means that we're only identifying regions that are known to contain protein coding areas, and those are exons, right? So as we will see in a few minutes, those are not the full sequence of the reads, but only exons and not the introns. Now, sometimes, especially in the situations where you want a very high confidence variant calling, you would like to sequence only specific genes. For example, if I'm looking at variants that I know of that are clinically relevant, and I don't care about anything else that is in the genome, I might want to only target specific genes, and that again is called amplicon sequencing that is focused on just an individual gene or maybe several genes or a gene panel. Now, how does sequencing actually work? The way it works, as I mentioned, is that genomic DNA is first shattered or cut up into pieces. Then those pieces are put into a library. There are adapters that are attached to those reads, and then it's put into a flow cell. Within the flow cell, there's a process called bridge PCR amplification. And so then all of this genomic material is fed into the sequencer, and the sequencer is actually trying to determine for each position of that read, what is that nucleotide? So as a result, you have this digital output, and that digital output is stored in several different file formats. These file formats that we're going to be dealing with a lot are FASTQ, FASTA, and GTF. FASTQ files contain the individual read sequences with the quality associated with each position. FASTA files typically contain just the full sequence. This could be the reference sequence, or even sometimes individual reads, or maybe portions like genes or something could be contained in a FASTA file. And then another file that is often used is a reference file, the annotation file, and that could be the GTF or the GFF file. So these three are going to be used a lot in our work with DNA sequences. And you really need to understand how these files are organized. So here you can see an example of a FASTA file. The FASTA file, as you can see, has a name. So right here on the top, this name could mean the specific gene, the specific chromosome, or the full organism name. Sometimes it will have the full description of that sequence. And then inside, it will typically have either a nucleotide or amino acid sequence without any breaks or organized in specific links. Like this one is organized in specific links. A FASTQ file contains information about sequencing quality. And so here you can see that for each read, like this is a single read, it will contain the sequence of that read, but also for each position, it will contain the information about the sequencing quality. And based on this quality, we can, for example, eliminate low quality reads or reads that are of inconsistent lengths. And we'll take a look at that today as well. So here's an individual read that, as you can see, contains 2 pieces of information. That's the same name. One contains the sequence, one contains the quality information. How do we analyze this kind of data? What we will use throughout this platform, throughout this program, is a platform called the T-Bioinfo platform. This platform is going to link the concepts that we use for each step of the analysis with the outputs that we're going to perform downstream analysis with. And this platform combines many different types of analyses that we will use throughout this program. And those will include alignment of reads to identify specific variants in positions according to their position on the reference DNA. They deal with variant calling, how to distinguish between random changes that are a result of maybe some changes that happen because of sequencing errors from actual variants and categorize them, as well as other downstream methods of analysis like phylogenetic analysis, mutability analysis, and others. So let's go back to the Learn portal, and I will show you how you will be able to get into that platform and get started with genomic data analysis. But before I do, please let me know if you have any questions about what we covered so far. You can just type in your question in the chat, and when we have a little break, I'll be able to answer any of those questions. So again, this is the program page where you registered, and then once you are registered, you should have created a profile, like Farhana already mentioned, and within that profile, you will be able to access different courses. So for example, the first course that we are talking about is Genomics. So this course will describe everything that I talked about today in greater detail and much slower. So if you want to repeat that, you can go ahead and do that. You need to be able to access all of the lessons that are here. And you will notice that some of the lessons have a yellow star next to them. So the first few lessons are going to be free. Anyone can take them. And then as you go into more depth, you will need to have a subscription. So that subscription is what is provided with this program. And thank you, Sanjay, for the question. Does the pipeline also include the suggested software and programs? Yes. So based on the pipeline, you will be able to download and install your own software if you want to rebuild the pipeline on your own. So what we'll do today is we will cover several of these examples just to kind of understand what are these different file formats, what do they look like, and how do we deal with them. And so as I mentioned, and by the way, does everyone have access? Can you please put in the chat if you have access to this course and you can access what I'm seeing on the screen? Again, I'm pasting the link in the chat. So please let me know in the chat if you're able to access this course and see what I see here on the screen. Yes, excellent, thank you. Okay, so we'll skip the first lesson. You can go through that on your own if you want to review the basics. Let's open working with DNA sequences in R. Okay, so don't worry, we're not actually going to install R. We're just going to understand the very basics of what's in a FASTA sequence, what does the DNA sequence look like, and what can we do with it. So as you can see right here, we have a brief introduction. And R, by the way, is a very useful coding environment that you can set up on your own system or you can run in the cloud. In today's session, we'll do a very short example. It's going to be very simple. You don't have to know any coding, but as you go through the different lessons, you'll learn more and more about what you could do once you learn about different packages that are included with R. The basic idea in R is that you create an object, and that object in this case is going to contain the sequence and can also have a name. So for example, string 1, you can put any string and you use quotation marks to identify what is a string as opposed to what you expect to be an object or a function. In string 2, I now have another string, and that string happens to be a DNA sequence. So to work with DNA sequences, I don't need any specialized tools if I'm just trying to load them, view them, or understand how many characters do they have. Sometimes we want to do very simple things like convert DNA sequences to RNA sequences. So here we just do a simple substitution. We substitute one type of a character, T, for another type of character, U. Sometimes we want to be able to use a library, and a library contains a lot of functionality that we don't have to write. We don't have to figure out how it works. It just works. And all we need to do is call a function with the right kind of parameters that will produce the result that we want. Now let's take a look at one of the most commonly performed tasks for genomic data, which is alignment. What is alignment? Alignment is essentially taking one string of sequences and trying to understand how similar is it to another string. For example, because it has many characters, I don't know how many of the characters are an exact match, how many were inserted, how many were deleted, or how many were substituted. So I would like to understand. And to do that, I don't need to think about manually aligning the sequence. I can use, again, a library. In this case, we're going to use two libraries, one called Decipher and one called BioStrings, to both perform alignment and visualize the results. Now, if you perform this, and first we're going to read about it, then we're going to actually do it together. As you can see, the result is going to introduce additional characters that are representative of gaps. So this sequence is longer, this sequence is shorter. So if I want to align them to each other, I need to add some additional characters that are gaps. Not only that, I will also see that if I introduce a certain number of changes, I can assign a score to those changes. Based on how many changes I have, I will have different number of scores as a result. For example, here we have 1, 2, 3, 4 missing nucleotides in the first sequence. So my score ended up being 102.68. In the second example, I have a much longer sequence in the first pattern, right? In the first sequence that I have. And then this is the second sequence I'm aligning, and my score is -206. So this gives you some intuition into what the score actually represents. It just tells you the quality of alignment. Now, in addition to that, we will see how this kind of simple alignment is useful in several different examples. And now when I click on next, so again, I'm on step number 3. Let's perform this. We can do this actually right in the browser. So all I need to do is I need to run this code. So by clicking on the run button, I am loading the libraries. Then I'm assigning different sequences to 2 objects. And then I am aligning those objects. And here, what's important for the score, to understand the score, I'm using the first one and then the second one, okay? After I click on run, it prints what the alignment ends up being. And so here in the console, I can see the alignment result. And the alignment result adds these additional characters at the end and provides me with a score. Okay, yes, you can definitely practice on RStudio. The reason why we're doing this in the browser is because the browser has the answers, right? So as you are getting started, you want to be able to replicate exactly what is expected. Once it works and you understand the syntax, you can take it onto your own environment and now work with that. But before you jump into that, let's make sure that you can get exactly what we have here, which also includes all of the libraries and dependencies that have to be installed and to work exactly the same for everybody who's using that. So that's why we're using the browser. Okay, so now let's try to see what happens if we change these sequences. Okay, so if you scroll here to the end, you can see that the second result is longer. Let's scroll through over here. Again, it is longer, right? Are there any packages? So the packages that we are using, Sanjay, is right here. The library, decipher and biostrings contains multiple functions. So to install these on your own system, you would have to install this library and this library. And you can do that in our studio. So I'll just show you in our studio. You will go to tools and you will say install packages. And here under install packages, you would have to install those packages. But I'm not going to spend a lot of time on this now because I'm trying to cover a lot in our first session. And after this, you will have plenty of time to set up everything on your own computer. That's why we want to use the browser. Okay. So here, the second exercise, again, all you need to do is just run it. And the objective here is to compare the scores, right? You can see the score here is 102. Here, the score is only 55, right? So the difference is noticeable, right? It's half. Now you have an assignment, and I'll give you just a couple of minutes here to perform this assignment. Now that you understand how this code works, and you understand that we're trying to align two sequences to each other, the assignment is to align, you can see here, the exact same sequence to each other, right? So here, we aligned DNA-seq1 to DNA-seq2. Then we changed a little bit the second sequence, and we again performed the same thing. Now let's try to see if you can figure out how to align the exact same sequence, DNA-seq2, to each other, to itself, sorry. Okay, so please try to do this and let me know when you have the result. Okay, so if you don't know yet, what are you supposed to do and how to do this? Let's do it together. What I will do is I will take a look at my first example, right? Always, if you ask anyone who's doing analysis, they always look at somebody else's examples. So let's just take a look at this example right here. The key line is line number 10. You can see that the alignment is performed in this line number 10. So I go to line number 10 and I see what is missing, right? Because if I run this, it is going to give me an error. So it says error running code. So let's clear this and let us write what we have here. So I can just copy this pairwise alignment DNA-seq1, DNA-seq2, paste it over here. But it said the assignment is, instead of doing it DNA-seq2, I need to change DNA-seq2 to DNA-seq1 to DNA-seq2. Let's try to run this. Excellent. So success running code, and answer is correct, right? And Sanjay and Raphael, you were asking how to run this on your computer, right? Obviously, this isn't something that you could have known that you've run correctly, because our system allows us to actually check on the back end that this is done exactly the same. Okay, so what is the score? The score that we got is 152.59. So let's compare the scores. Here we have 102. Here we have 55. And here we have 152. Let me ask you a question. What do you think happens to the score as sequences become more and more similar to each other? Does the score increase or decrease? Exactly. So Raphael correctly said, and Kome, the score will increase, right? So we can see that the sequences are exactly the same. The score is higher. And when we introduce more and more changes, it is going to be different. Why do you think it is different? So as you can see, you probably just copied this and pasted it here. But the assignment is, right, read this, please. Alignment between the exact same sequence. Okay, so if you just take this, which is DNA-seq1 and DNA-seq2, you're going to get a result, right? But the result is going to be just like here, 55.4, 55.4, right? And the answer is incorrect. So what you need to do is you need to change this to 2. Does that make sense, Sanjay? Excellent. Okay. So very quickly, we understood how to work with DNA. We know what we're looking for. We know how to evaluate it. We can align multiple sequences to each other. As you go through the material, you will find more and more advanced steps that you need to take. And that will give you a good sense of what people do when they use bioinformatics. Now, as you remember, this individual sequence is only one read. So let's take a look at how does this data actually look like when we have actual sequencing raw data. To do that, we will go to the platform. So the platform is called the T-Bioinfo platform. You also have a lot of introductory material on the platform. And what we will do is we will perform alignment of many, many different short sequences to the reference genome. Okay, so please let me know once you reach this platform and log in. How do you know that you're logged in? You hover over this area right here, and you should see your email address right here. So if, and please confirm that you can log in, because sometimes people don't receive the email and password. Typically, your email and password from the Learn portal should work here as well. But maybe you didn't get it or you don't remember it or something. So please do let me know that you can log in. Okay, does anyone have access to this platform? Okay, so you don't have it. So if you go here, right, you can just log in. So I'll log out. Okay, you go to log in, and you use the exact same password that you have on your Learn portal. Do you have that information? Okay, Sanjay, it looks like you do have an account. Okay, perfect. So what we want to do today is going to be a demo. It's not going to be an actual analysis. An actual analysis will take some time, but in this case, we're going to go through a very brief example. Right here, you can find all kinds of sections related to next generation sequencing data. What we want to do is we want to go to mutation variant, parallel analysis of mutation variant data, and click right here on the drop down. Okay, Arpita, could you please put your email address so we can take note of it? Farhana, if you could maybe take note, so we can share that with Sonarika. And by our next session, or hopefully even before that, everybody should have access. So you should be able to get your password. All right, excellent. So looks like you're okay. So it looks like you're at LSU. So please email Farhana, or Farhana, I don't know if you can put your email address here, but also you can email support@pine.bio, and you should get your account information there. Okay, so what we want to do is we want to go to this section, mutation variant, parallel analysis of mutation variant data. Okay. click on the drop down right here, and you want to go to this first demo. So again, this is going to be a demo variant calling cell lines. Once you're in, you'll see that each button here represents an algorithm or a script. Just like what we did there in R, that's a script. I can make it into a standalone script provided with some inputs, and it's going to run every time I call it. So here I have many different scripts, and my goal is to piece them together into a series of scripts that we will call pipeline. These pipelines can contain from 2 to maybe 20 or even 50 different scripts combined together, and they will provide us with an output that is more easy to work with than the individual intermediate outputs that we could produce just running those scripts. So to begin, you have to click on Start, and every time you will have a pop-up that has some explanation of what the script does or what we are intending to do in this specific pipeline. Then click on OK, and you will have two options highlighted. These are the two options that you have to select next. Either you would like to clean your data, or you would like to align your data to the reference genome, which is called mapping. So this is mapping of those reads to the right position on the reference genome. Let's click on Bowtie 2. So Bowtie 2 is an algorithm that is used, it relies on the Burroughs-Wheeler transform, which is a method of transforming strings that is used in many different applications, including search. When you do a search and you want to find a specific word in your document, this is also an efficient way to search for that word. So when you work with strings, these algorithms are multi-purpose algorithms. And in this case, this particular BWT was used in Bowtie. And if you want to know more about this algorithm, you can just go to this publication right here. Once you click on Bowtie 2, you then want to add this module for visualization. So what we'll use is, again, a standalone set of functionality called JBrowse, which is a package that is used to visualize the genome. And you can find it being used in many different parts of the internet. If you've ever looked on reference genomes and you've kind of seen some compilations of data, likely they have used JBrowse to visualize that. So now we're going to click on End, and we have ourselves a pipeline. This pipeline has 4 different steps. We loaded the data in Start. We then map the data to the reference genome, and then we want to visualize the individual reads that have been mapped on the genome position, and then we finished our pipeline. Okay, so if you completed this exactly like I did, you should have gotten this link right here that says visualization. Okay, did anyone complete this pipeline and get the link for visualization, or should we try to do this again? All right, perfect. So Raphael got it. Did anyone else? All right, excellent. So let's click on visualization and let's see what comes up. Yes, and I will go back really quickly just to repeat everything from the beginning. So we're on areas of analysis. Here, you can see that we have mutation variant, parallel analysis of mutation variant data. And I click on this drop down. Okay, so Raphael, we'll get to the visualization in just a second. Then you click on this first demo variant calling cell lines. Once you get here, you click on Start, Bowtie 2, visualization, End. Okay, and now you should have a link that says visualization. Now, initially, when you just click on visualization, you might see something like this. There's nothing there. Okay, everything is empty. You can do 2 things. First of all, this is a full chromosome, right? So right here, you have a drop down that has different chromosomes. Right here, you have the exact position on the chromosome. Now, I don't just know the position for every gene. So what's a better way to do this is to search for a gene. For example, a gene that I know is BRCA1. So BRCA1 right here is a gene on chromosome 17. So I can click on Go. And now we found this gene, right? So you can see that BRCA gene actually has many different transcripts. And what you can see is that the pink area is the exon and the black line are the introns. So it consists of many different exons. Some of them are very short, some of them are longer, and a lot of introns. And that is your genome gene. So this right here is the GTF file, the genome annotation file. That's right here, genome genes. Here you also have the reference sequence. The reference sequence is the actual genome. So if you zoom in and you want to see the sequence, this is the sequence right here. So I have to zoom in a lot, and then you will see your sequence as well as those codons that we were talking about, right? And if you want, you can actually move this to the top. Okay, so now that we have this, let's zoom back out. We don't actually want to see the reference sequence. Right now, it doesn't make sense for us to look at that close of a zoom in. We want to see our exons, and we want to see what the gene looks like. Okay, so the chromosomes are right here. The position of the chromosome is right here. This is the annotation. All of this has nothing to do with the data that we uploaded. What do you think the data that we upload looks like? Well, that data has individual reads, right? So right here, you can see that we actually uploaded 2 data sets or 2 files. These are 2 FASTQ files, and they are represented by the aligned file. So now the FASTQ has turned into a BAM file. And if you click on one of them, just let's click on the first one called alignment. You can see down below, it says too much data to show, zoom in to see detail. So let's zoom in a little bit. Let's scroll down. And now you should be able to see the individual reads. Okay, so in fact, if you click on them, you will see all that information that we saw in the FASTQ file. This is the actual read. And this is the sequence and its quality. You can also see how long is this read. So if you found a read, tell me how long it is. If you looked, if you were able to open it, you should be able to see in this information of an individual read, how long is the read. Okay, or you can see it on my screen. So how long is this read that I'm looking at? Where did I clicked on one of the reads. So these are these little, yep, exactly. Thank you, Arpita. So if you click on one of these, this window will pop up and you will see the read length, right? And so that previous one was 101. This one is 108. So you can see that the read lengths are actually a little bit different, but they're all around 100 nucleotides long. Okay, 108. Yeah, so it really depends on which read you selected. And it doesn't matter which one, right? This is just to show you that these individual reads are right here. And those are the sequences. Just like we were aligning those 2 sequences to each other, that's what happened here, but with many different sequences. Now, this information, as you can see, is cut off. So we actually don't see the full information that we have. So instead of looking at this at the level of alignment, we can also select coverage and SNP. Okay, so if you scroll down right here, you can see that not all regions are covered equally, right? So since this is kind of shorter than the other one, I'm again going to move it up. Okay, and now I have a question for you. Is this whole genome sequencing, whole exome sequencing, or amplicon sequencing? What do you think? Amplicon. So Arpita is saying amplicon. Any other ideas? Arpita seems like exome, Sanjay is saying. So Arpita, why do you think this is exome? And Sanjay or Kome, why do you think this is exome or amplicon? Yeah, why do you think what you're saying? Okay, the answer is that it's not full genome, right? Okay, so Arpita is saying sequence stretches are short, only coding area seems to have the reads. That's right. So if you look where this is, it is only the coding region, right? So this matches exactly this red or pink little section, as well as all of these abundances. So the majority of the reads actually just map here. How do I know that this is not just amplicon? The answer would be if I can find another gene, right? So first of all, I can of course zoom out and I can see that multiple genes have coverage. So this will take a little bit to load, but you will see that multiple genes have coverage. But also I can go to another gene. Let me just go to another gene, for example, TP53, the P53 gene. Go to this one. Go. Right, and here again, I see that the abundance is just around the exons, right? So because I can see that the abundance in multiple locations are just above exons, I can conclude that this is whole exome sequencing data. Apart from this, you can see that this section is supposed to show me SNPs, and those SNPs are the actual variants, right? The changes in the sequences that we will find that are associated with some phenotype. And in this particular case, we don't see any changes in the sequence. But if we keep scrolling, eventually we will find them, right? So we're at the end of our session. So let me briefly conclude. What we saw today was how to analyze next generation sequencing data and find genomic variation. This genomic variation is going to be the basis of a lot of what we do throughout this program. We're going to look at multiple sequence alignment and alignment to the reference genome. We're going to understand the relationship between nucleotide sequences and amino acid sequences. And we're going to use a variety of tools that first we need to understand the logical application of these tools. How can we build multiple types of pipelines to get what we want from the type of data that we have? This will include calling somatic mutations, like in cancer. It will be performing genome-wide association studies and identifying what particular positions on the genome can be associated with a phenotype. We're also going to look at how different types of changes have an impact on the structure of that particular protein. And all of this will be provided to you as a series of practical steps that you can perform independently over the course of the next 3 months. So Rafael, can you download the pipeline file from TBio and use in RStudio? Most of these methods are not written in R. You can perform some of the same in R, but not all of it. So you can't just download and do it in RStudio. Some of the methods that we will use, you can. And so when we get to those, I will specifically point those out. Now, you will have multiple examples that show how this type of analysis is used for different types of applications. So in the same course that we were, the synchronous course, You will go from the very introductory material that we looked at today to other types of analysis techniques, multiple sequence alignment. You will find exercises that you should be able to do in R as well. Analysis of mutability, differential mutation analysis, copy number variation, and others. And so as you go through this course, hopefully you will be able to appreciate the value that is found in genomic sequencing data and some of the baseline experiments that you can analyze data from to understand the differences between mutations, variants, differences in copy number, variation, etc. Can we perform our own set of experiments on these platforms and use the pipeline? Yes, you can. And that was the point of that project that we discussed when Farhana was showing you the profile. So this is an advanced program in the sense that it will provide you with skills that you can directly apply to a data set. Do we have courses on methylation analysis? Yes. So whole genome bisulfite sequencing is a part of epigenomics. So you will find that in a course called epigenomics. Okay, so if you have any other questions, please feel free to reach out to us. Farhana is at LSU. You can meet with her. We want to know what questions, what concerns, what issues you are facing so that we can prepare for our next Q&A session that is going to be February. When? February.
The Q&A session, the first one is February 21st, and the second one is March 18.
Okay, perfect. So those are the dates. Any questions that you have, please make sure to let us know about them. If you have any questions about, what else is covered or what else are we going to be able to do, you can go back to the syllabus and refer to the different sections that we have. We will be sending you videos, recorded videos that you can watch that seek to connect the different aspects of what is found online. And if you have any questions during the Q&A session, we will be able to address those. All right, and excellent, Rafael. I'm excited to see that you're enjoying it. So hopefully everyone is going to benefit from this as well. Finally, my personal email address is ilya@pine.bio. I can reply if you have any technical issues. And if you have any technical issues about any of the assignments, you can also write to support@pine.bio and get any questions from our support team answered right away. Thank you again for joining today. We're going to be excited to see your progress and looking forward to the next few months together. Thank you, everyone. Sorry.
Thank you.